السلام عليكم ورحمة الله وبركاته. Hello everybody, how are you? Hope you are all well. I am Dr. Karim Metwalli, lecturer of histology. In this video we are going to continue by the second part of the integumentary system, skin and its appendages. What are our ILOs? LOs for this lecture. By the end of this lecture, you should be able to describe the structure of the derms, differentiate between the papillary and reticular derms, enumerate the types of the skin glands, describe the structure and correlated functions of the species glands and types of sweat glands. Describe the structure and correlated functions of the hair follicle and its associated structures. Describe the structure of the hair and the hair follicle. Let's continue with the dermis. The epidermis is the underlying structure under the epidermis. It is the deeper part of the skin, and it is separated from the epidermis by the basement membrane. It is mesodermal in origin, and it is formed of connective tissue rich in blood vessels, lymphatics, and nerves. Its thickness varies in different body regions. This is the dermis, and this is the epidermis. This is the dermis. The dermis contains epidermal derivatives or skin appendages like hair follicles, species and sweat glands, and it is divided in two parts. The superficial part, which is the papillary layer, and the reticular part or reticular layer, it is the deeper part. And there is no clear boundaries between the two parts, the papillary and the reticular. The papillary layer is a thinner superficial part of the dermis, and it forms the dermal papillae. This part is the dermal papillae. The dermal papillae are the protrusions of dermal connective tissue into the epidermal layer. The superficial part, as we mentioned, formed of loose connective tissue, so it contains more connective tissue cells and less connective tissue fibers. It contains capillary loops and also contains touch receptors, which are Meissner corpuscles. The connective tissue cells like fibroblasts, mast cells, macrophages, and other leukocytes, and connective tissue fibers, collagen type 1 and 3 fibers. The reticular layer, which is the thicker and deeper layer, is formed of dense regular connective tissue with collagen fibers, thick elastic fibers, reticular fibers, forming network, so it is called reticular, as it is in the form of reticulum. This layer is more fibrous than cellular, and the fibers are coarse, but it is less vascular than the papillary layer. This layer also contains receptors for pressure called Pacinian corpuscles. P for P, Pacinian corpuscles for pressure, sensation. The dermis contains blood vessels and blood capillaries that provide nutrition to the non- or avascular epidermis, in addition to its thermoregulatory function. Many arteriovenous shunts are present, decrease blood flow to minimize heat loss in cold conditions, and vice versa. The subcutaneous connective tissue, or hypodermis or superficial fascia, it is not part of skin, and it is formed of loose connective tissue rich in adipocytes or adipose tissue, and it has extensive vascular supply. What are the types of skin? Skin have two types, the thick skin and thin skin. The thick skin is found in palms of the hands and soles of the feet, while most of the body is covered by the thin skin. What is the difference between the thick skin and thin skin? In epidermis, the epidermis for thick skin formed of the 5 layers, and it appears thick with thick stratum corneum. The keratin layer, it is thicker than that of the thin skin. The epidermis in thin skin formed of 4 layers with no stratum lucidum, with thin stratum corneum. The all thickness is thinner than that of the thick skin. While the dermis, the skin appendages like hair follicles, species glands, and rector bile muscle, they present mostly in thin skin while absent in thick skin. While sweat glands are more abundant in the thick skin and less abundant in the thin skin. As we see in these two pictures, the difference between the thick skin and the thin skin. The epidermis in the thick skin is thicker than that of a thin skin, thickness of the layer as we see, and the keratin layer thicker in thin skin than that in thin skin. The skin appendages, as we see here, the hair follicles and species glands, rector bile muscle, present in thin skin and not present in the thick skin. But sweat glands, this is the sweat duct. Sweat glands are more abundant in thick skin than thin skin. Again, let's know the difference between the thick and thin skin. The thick skin found in palms and soles, while the thin skin cover the body surfaces all over the body except palms and soles. The surface of the thick skin has fingerprints, while that of the thin skin has no fingerprints. As we see in the pictures, the epidermis of the thick skin is formed of the 5 layers, described before, while that of thin skin formed of only 4 layers with no stratum lucidum. The dermis of the thick skin contains no hair follicles, no species gland. There are numerous sweat glands. The dermal papillae are high and numerous. While in the dermis of the thin skin, the dermal papillae lower and fewer in number than that of the thick skin. They have the thin skin has hair follicles, erector bile muscle, sebaceous glands, and sweat glands, but sweat glands are less than that of the thick skin. As regards the pigmentation of the skin, the thick skin is less pigmented than that of the thin skin. The thin skin is more pigmented, while thick skin less pigmented. Now we are going to discuss skin appendages. The skin appendages include skin glands, sebaceous and sweat glands, the hairs and hair follicles, and the nails. The hair and hair follicles, with its sebaceous gland and erector bile muscle, are called pilosebaceous unit. Let's start with skin glands. The human skin glands include sebaceous gland, which is related to the hair follicles, and sweat glands, which are coiled tubular glands and include two types, eccrine sweat glands and apocrine sweat glands. Let's start with sweat glands, and we are going to discuss sebaceous gland with the pilosebaceous system. Sweat glands are of two types, eccrine sweat glands and apocrine sweat glands. Eccrine sweat glands mostly open into the surface of the skin, while apocrine sweat glands open their ducts open into the hair follicles, the upper part of the hair follicle above the opening of the sebaceous gland. The sweat glands are simple coiled tubular glands present deep in the dermis all over the body, except in very few sites, like lips, glands, penis, and nail beds. They are particularly numerous in thick skin. Sweat glands are commonly classified into two types, eccrine and apocrine sweat glands. The eccrine sweat glands are the common type in man, and they secrete by exocytosis. Merocrine mode of secretion, and in a cross section under the microscope, the eccrine sweat glands consist of two parts, secretory and excretory part or duct. The secretory part, it is a highly coiled tubule situated deep in the dermis or the hypodermis, or what's called subcutaneous tissue, which appears, and this secretory part appear in cross section or oblique section as little clusters of acini. These acini are lined by two types of cells and surrounded, and these cells lie resting on basement membrane and surrounded by myoepithelial cells. The two types of cells are cuboidal, and they are dark and clear cells. The excretory parts or the ducts, these collect the sweat secretion from the secretory acidic, and they run in a spiral or tortuous course in the dermis and the epidermis to reach the skin surface. Thus, it has two parts, the dermal and epidermal parts. And in the epidermis, they are lined by stratified cuboidal epithelial cells, two layers. And as it passes through the derms, and in the epidermis, it follows a helical course and is aligned by the keratinocytes. It opens on the surface by the sweat pores. Cells reabsorb some electrolytes and excrete others as urea and lactic acid. They are ion transporting cells. Okay, let's now discuss the secretory unit with the lining cells. The first cell, dark cells or mycoid cells. By light microscope, these cells appear as inverted cones or inverted pyramids with their broad ends directed toward the lumen of the acini, and the narrow ends directed to the opposite side, directed to the basal lamina, but they don't reach the basal lamina. The nuclei of these cells are dark. By electron microscope, the cytoplasm of these cells contain rough endoplasmic reticulum, Golgi apparatus, mitochondria, secretory granules, and these cells secrete glycoproteins, mucus-rich substance. The other type of the lining cells of a secretory unit are clear cells. The clear cells by light microscope appear as pyramidal cells with narrow apices directed toward the lumen, while the broad or expanded base extending to the basal lamina. By electron microscope, the cells show a basal plasma membrane enfolding, and so these cells have the character of ion transporting cells. There are also intercellular canaliculi. The cells contain abundant glycogen, and so these cells appear clear and pale. There are no secretory granules. The cells are concerned with the secretion of water secretion into the intercellular canaliculi. They have similar organelles as the dark cells, but there is no secretory granules. The acinar cells of the secretory units are separated from the basement membrane by the myoepithelial cells, which are rich in actin filaments, and their contraction assists in expelling the sweat secretion and discharge the secretion into the ducts. The other type of sweat glands is the apocrine sweat glands. The apocrine and sweat glands are less numerous in human than in other mammals, and are only present in certain sites like axilla, areola of nipple, perianal region and the pubic region. They start secretion at puberty under hormonal control, and they are also merocrine glands. The epocrine sweat glands are less numerous in human and are present in limited sites, as we mentioned, like axilla, pubic region, groin, and areola of the breast. They are merocrine gland, secrete through exocytosis, although they named epocrine gland. The secretory part similar to the other type, but has wider lumen, and it is lined by a single layer of cuboidal cells and surrounded by myoepithelial cells. The duct is coiled and opens, as we see in this diagram, opens into the hair follicle just above the opening of the sebaceous gland, and not into the skin surface. The duct lined by two layers of cuboidal cells, and the epicrine sweat glands secrete a viscous secretion under conditions of excitement or aroused emotions at puberty, and the secretions has no odor, but when acted upon by bacteria, it becomes degraded into odoriferous products. Has a special odor. The secretion of epicrine sweat glands is under the influence of hormones and begins after puberty, as we mentioned. Now let's know the structure of the species unit. The species unit formed of the hair and hair follicle, species gland, and the erector pili muscle. This is the species unit, which is formed of the hair follicle, species gland, and the erector pili muscle. The structure of the hair. The hair is cornified epithelial thread. It is found anywhere thin skin is present, except in certain sites, like red margin of lip, glans penis. In longitudinal sections, the hair is formed of the shaft, which is projecting above the surface of the skin, the root, which is embedded in the skin, and it is surrounded by the hair follicle, and finally the hair bulb, which is the terminal dilatation of the hair, which is invaginated by vascular connective tissue mass from the dermis, called hair papillae. Okay, again, as we mentioned that the hair is formed of the shaft projecting above the surface of the skin, and the root embedded in the skin surrounded by the hair follicle, and the hair bulb, which is the terminal dilatation of the hair, which is invaginated by vascular connective tissue mass from the dermis, called the hair papillae. Let's now know the structure of the hair shaft. From inside outwards, it consists of three parts. the medulla. It is the central core, as we see in the picture. This is the medulla, the central core. The medulla formed of vacuolated cells contains soft keratin, unlike that of the nail, the soft keratin like that of the epidermis. As there is two types of keratin, soft and hard keratin. This also, the medulla also contains melanin, contains no melanin, where it is absent in the fine hair. In fine hair, there is no medulla. The cortex, outer to the medulla, it surrounds the medulla, formed of flattened non-nucleated cells, and contains hard keratin and also melanin, which color the hair. The cuticle, it is the most outer part of the shaft, and it is continuous with the cuticle of the root. It is formed of scale-like non-nucleated cells, and also it contains hard keratin. Okay, what are the hair follicles and what the structure of the hair follicle? The hair follicles are a tubular invagination of the epidermis that surrounds the hair root. It develops as down growth of the epidermal cells into the dermis, and the deepest part of the epithelial down growth forms a cluster of rapidly dividing cells called germinal cells. Germinal matrix in this part, as you see here, this is the matrix containing the germinal cells. These cells are responsible for the germination of the hair. Okay. The cells from the germinal matrix form the concentric layers of the hair shaft as they move toward the surface. Thus, the root of the hair has a structure similar to the shaft, but the root surrounded by the hair follicle in this part. Okay, so again, the root formed has a structure similar to the shaft, but the root surrounded by the hair follicle. Both the roots and shaft germinates from the germinal matrix, the matrix which fitted over the dermal papillae. Okay, let's now know the structure of the hair follicle. The hair follicle formed of the external or the outer root sheath, this part, and the internal root sheath or inner root sheath, this part, and both layers, the internal and the external, are surrounded by connective tissue sheaths or dermal sheaths derived from the dermis, form outer covering to the hair follicle by condensation of the surrounding connective tissue. This part is the connective tissue sheath. Okay. The external root sheath or outer root sheath, this part is the epidermal down growth that connects the germinal matrix to the surface. This is the epidermal down growth, the outer root sheath. As it develops as down invagination, then they becomes canalized near the surface of the skin, and it shows the all layers of the epidermis of the skin, but deep down it becomes thinner and loses some of the superficial layers. Near the bottom of the follicle, the matrix here consists only of the stratum germinativum. Okay. The glycine membrane, it is the second basement membrane that separates the hair follicle from the dermis. The glycine membrane is the basal lamina of the epidermal cells, epidermal epidermis, down invagination, surrounded by basal lamina. This basal lamina becomes thickened and called glycine membrane, and this membrane separates the hair follicle from the dermal sheath or the dermis. This layer it is non-cellular and hyaline. Okay, let's see this diagram or photo in cross section of a hair follicle and root of the hair. As we see, as we mentioned, that the root has the same structure of the shaft, so the root shows the medulla, the cortex, and the surrounding outer the root cuticle. Then the internal root sheath and the external root sheath and the connective tissue root sheath or the dermal sheath. Okay, this all the wall of the follicle. In longitudinal section, as we see here, this is the root formed of the medulla surrounding by the cortex, and then the cuticle. This is the root, then become the shaft near the surface. Okay, this part, the root or the shaft, then surrounded by the internal, this part, the internal root sheath, then the external root sheath, then the connective tissue sheath of the hair follicle or the dermal sheath. Okay, let's know the structure of the internal root sheath. It is the inner most layer of the hair follicle, and it is formed of three parts or three layers. The outer part, which is under the outer external root sheath, called Henel's layer. The Henel's layer, it is extension of outer brachial layer and formed of a single row of flat cells. The next layer is the Huxley's layer. It is the middle layer of the internal root sheath, and it is an extension of a granular layer formed of one or two rows of cells and contains trichohaline granules. The inner layer of the internal root sheath, which is continuous with the cuticle of the hair. It is also the cuticle of the internal root sheath. It is an extension of the horny layer and formed of cornified scales. The dermal sheath formed of the dermis, as we mentioned, as it is a dense connective tissue sheath surrounding the hair root. And this part is the hair papillae and the germinal cells in this part, and this part called the matrix of the hair. Okay, again, the structure of the hair and the hair shaft and follicle. This is the hair shaft projecting above the surface, and this is the root. The root surrounded by the hair follicle. The shaft formed of inner medulla covered or surrounded by the cortex, then the hair cuticle. The root has similar structure to the shaft as we see here, medulla, cortex, and the outer hair cuticle. And the root surrounded by the hair follicle. The hair follicles develop as down growth from the epidermal cells to the derms. The hair follicle, as we see, formed of the from inside outward internal root sheath. This part is the internal root sheath with its three layers. Then the external root sheath, which is formed of the epidermis and epidermal cells. All layers then becomes only formed of some layers, loses some superficial and deeply become only formed of the stratum germinativum with the matrix here. Then the outermost covering, which is the dermal sheath or connective tissue sheath, and it is derived from the dermis covering the hair follicle, and it is separated from the external root sheath by the glycine membrane, which is non-cellular hyaline layer. And it is considered the second basal lamina or basement membrane of the dermis. This is a transverse section or cross section through the hair follicle and the hair root by the light microscope. As we see here, this is medulla and cortex surrounded by internal root sheath, and then the external or outer root sheath, then the dermal sheath of the dermis. Okay, the hair bulb. Let's now know the structure of the hair bulb. The hair bulb is the terminal dilatation of the hair follicle. This part is the hair bulb. It is formed of cellular mass at the bottom of the hair root sheath and root, the hair matrix. As we mentioned, this part contains the hair matrix, and it is responsible for the hair growth. It receives nourishment from the hair papillae connective tissue, as it is highly vascular, containing blood vessels responsible for nourishment of the hair and these cells. We mentioned before that the pielospecies unit includes the hair and the hair follicle, species gland, and the erector pili muscle. So let's know the structure of these species glands. The species glands are developed as the outcross from the hair follicle, as we see here. It is a simple branched alveolar gland formed of secretory part and excretory part or ducts. The secretory part lined, as we see here, by stratified cells, more than one layer. The basal cells are flat. These cells, the basal cells are flat, and they proliferate, accumulate fat droplets, and they accumulate fat droplets, and then lose their nuclei. Then these cells burst and liberate its contents as a type of holocrine secretion, holocrine. The cells burst and liberate its content. What about the duct system? What about the structure of the duct? As we see here, this is the species gland formed of secretory part and duct system. The duct, as we see here, is short and wide, and opens into the upper third of the hair follicle. In the external genitalia, it opens directly into the skin, not into the hair follicle. Okay, again, the duct, and this is the secretory part in the form of alveoli. And the secretory part lined by stratified cells with basal flat cells that proliferate, and as they proliferate, accumulate fat droplets, and the cells then burst and liberates its content as a holocrine type of secretion. The duct part, as we see, again, the duct short and wide and opens into the upper part of the hair follicle. In external genitalia, as we mentioned, it opens directly into the skin. What are the functions of the sebum? What is the importance of the sebum? Sebum oils the hair and also lubricates the surface of the skin, and also has antibacterial action. Sometimes there is an obstruction of sebum secretion, and with overlying bacterial infection. This results in chronic inflammation of species gland and hair follicles in a clinical condition called acne. And the last part of the pielospecies unit is the erector pili muscle, which is a small bundle of smooth muscle cells extending from the fibrous sheath below the level of the species glands to the dermal papillary layer. Its contraction pulls the hair into an erect position, so it is responsible for erection of the hair during sympathetic stimulation, and also when it contracts, it squeezes the content of the sebaceous gland. The first question, the keratinization process in the epidermis occurs in about 2 to 6 hours. The keratin fiber formed in this process is called soft keratin, in contrast to the hard keratin of the hair and nails. This process includes A. Transformation of the basal cells into the brittle cells, or B. Transformation of the brittle cells into the granular cells, or C. Transformation of the granular cells into the cornified cells, or transformation of the bone marrow cells into Langerhans cells. As we mentioned before, that the epidermis is formed of the 5 layers, and the basal layers of the epidermis, which is the stratum basale and the basal part of the stratum spinosum, are responsible for the regeneration of the epidermis. So the cells undergo structural changes as they pass through the epidermis from the basal layer to the keratin layer or stratum corneum. So the answer is the transformation of the granular cells into keratinocytes, transformation of the granular cells into the cornified cells. And this transformation of the granular cells into cornified cells, when granular cells are transformed into cornified cells, this involves a breakdown of the nucleus and other organelles and the thickening of their plasma membrane. Okay, so the answer in the keratinization process, there is a transformation of the granular cells into the cornified cells. The non-membranous basophilic granules containing proteins, which is responsible for binding or aggregation of the keratin filaments. The keratohyalin granules, lamellar granules, or melanin granules or neurosecretory granules. Exactly, it is the keratohyalin granules, number A, as these granules contains proteins, and these proteins are responsible for aggregation of keratin filaments. As the number of these granules increases in the cytoplasm of the granular cells, the contents of these granules released into the cytoplasm of the keratinocytes of the granular cells, and these proteins start to aggregate keratin filaments into tonofibrils, and thus initiating the conversion of the granular cells into the cornified cells and the transformation in the keratinization process. Which layer of the hair follicle contains actively dividing cells responsible for hair growth? A. Cuticle, B. Cortex, C. Medulla, D. Matrix. Okay, exactly, it is the hair matrix, as we mentioned, that it is contain rapidly dividing cells called germinal matrix or germinal cells or matrix hair cells, which germinates the hair. So the matrix is responsible for the hair growth. Okay, thank you so much for your listening.